Okay, I made you a co-host.
Okay, cool.
All right, so we just had a meeting with Lauren, and she wants these uh, primers to be ordered soon.
Hmm.
So for now, we don't know the exact sequence of the vaccine mRNA. I don't know if it's actually proprietary. So I was trying to look through these, FDA files and see if anyone has disclosed the sequence, but I don't have time for that now. So for now, what I'm doing is I'm just taking the SARS-CoV-2 sequence and designing primers and probes for that, and we expect that it should be able to bind to the vaccine
Right. Okay.
sequence as well.
So where were you going to look that you couldn't find it?
So I think WHO has a report.
Oh, okay, okay.
So if you Google vaccine candidates WHO, it took me to some page, and they have some, before they get authorized, they have their provisional names, which have like numbers mRNA 1739, something like that. Sometimes on those they'll have these patent documents. So for example, this is the whole thing from WHO. Yeah, and they have a few links to clinical trials. I don't know where to find this stuff, so just looking all over.
Okay, okay. I thought there might have been some like, I don't know, central repository for this kind of information that you're looking at, but I guess this is just for this situation, right?
Yeah, I think they make it harder for you to find more details about their stuff. So yeah. So for now, what I did is long time ago I grabbed this sequence from NCBI. I think it's the first human that was sequenced who had the SARS-CoV-2 or something. So that's why it's U1. And from here, I found an S gene, which is the surface glycoprotein,
Right.
or also called spike protein. So I just copy pasted that into another sequence file on Benchling.
Okay.
Okay, so I found a couple of papers that were doing this stuff. So I just search for S gene here, and I go to their supplementary and copy their primers. Yeah, so they mentioned forward and reverse. So I just put them on to this thing here. There are these. Alright, so first thing you would do is to look for literature, find any primers, but once you find them, you have to verify that they are working well. So once you paste these two sequences, you will right click the region between them and say create primer pair. I think it's already created. Okay, so it appears, so they have some shortcut buttons here on the right.
Mhm.
This one is for primers, one with forward and reverse. And they have individual primers as well as pairs of primers. So if you go to a pair and then ask it to show you secondary structures, it will open another window here, and it runs some thermodynamic algorithms, and it shows you what would be the free energy of binding, and you want to make sure that this energy is low, not low, as in it's closer to zero. It should not be -5 or -10. That means the binding is very strong.
Right. Okay.
Okay, so you just click on one of these and it shows you what structure it's using to calculate. So this is the structure with the strongest binding.
Okay.
I
So,
generally try to avoid nucleotides multiple 3 or 4 that are continuously binding to each other. But if the energy is not too low, it should be okay. So -2 is decent. If it's -5, I would worry more. -6.
right. So is there like a balance you want to strike between strong and weak?
No, so you want this primer to bind only. Where is this? Okay. So this primer you want it to bind to the uh, target sequence.
Right.
So this energy it is showing you is for the primer to bind to itself. So if it binds to itself, then it's not available to bind to the target.
Oh, I see. I see. Okay.
Yeah, so what it's showing you is the blue is one set of the primer, the red is another of the same molecule. So two
Mhm.
things of two different molecules of the same primer, if they bind, then it's called a homodimer.
Right. And you don't want that, right?
You
Yeah.
don't
Okay.
want that.
Yeah. Okay.
And if the binding of this thing is very strong, then because it won't bind to the actual template.
Right. Okay.
And the other thing is monomer. So this thing right now it's not showing any binding here, but if it's a single molecule that bends onto itself, it forms what we call as a hairpin. If that forms, that's a lot more likely because it's a unimolecular reaction. It doesn't require two molecules to come together.
Hmm.
So this is definitely you want to avoid this as much as possible.
Right.
So whoever designed it for this paper, I think they did some background thermodynamic check. In few papers, I see that the primers they use are not very efficient. They still work, but for ddPCR, I've noticed that it's a lot more stringent. It seems like compared to qPCR.
Hmm.
I don't know the reason behind it.
Right. Okay. So you said you try to avoid this in the middle here where you have four base pairs in a row that are binding.
Yeah, so if I'm trying to be stringent, I usually avoid that. Also, if it has G's or C's here, it's a little more uh, stronger binding. So you can see the energy here is -4, whereas in the left one it's only -2, because here it's only A's and T's.
Okay.
But I've been told that I should not worry too much about this if the energy is not too low.
Okay. And so for
So
like
if they
rule.
are anywhere close to the end, sometimes I manually trim the primer, so I remove those bases that are problematic. But if it's dead in the middle, then it's hard to tune it without. So I can reduce the last 7 bases here, 1, 2, 3, 4, 5, 6, right? And then I have to include maybe 7 more on that side to maintain the length similar.
Mhm. Okay. So did you say like -5 was kind of your cutoff for like the?
Yeah, so -5 is the regular cutoff.
Mhm. Okay.
You can be a little more lenient and go until -7
Okay.
for the dimer, but for monomer, I think -3 would be the limit.
Okay.
If possible, I absolutely avoid any hairpins, but it's very hard. Sometimes the regions have GC content that's very high or low, then it makes you hard to design primers.
Right. Okay.
One more thing we need to look at is the difference in the melting temperature between the two primers should not be too high. So to look at melting temperature, if you select any sequence on Benchling, it shows you on the bottom the GC content and the melting temperature. So if I select this primer, it shows the melting temperature for this binding of this thing to the complementary sequence.
Okay.
So this is 58 point something, and the other one is 58.2. It's showing only one decimal, right? So the difference is less than a decimal, so it's decent actually.
Okay.
And I think the guidelines generally say it should be within 2 or 3 degrees.
Okay.
I mean, you can make it work even if it's farther apart. Just the efficiency of the qPCR might be low or something. But I've always amplified stuff with qPCR. It's
Yeah.
not a issue.
Okay.
And the other thing we look at is this heterodimer. So if you put two different sequences, like this guy can also bind to that molecule, the forward
Mhm.
and reverse. So that's in the heterodimer. If you open that, it's showing similarly for nucleotides. I think this can be ignored as well.
Okay.
So this thing that I got from the paper, it seems reasonable so far.
Right.
So I named it something here, and then I'm looking at another paper. So I put in those sequences somewhere else. And so for this one, I'll do again. So I copied the sequence from there. Let's say you find the sequence in this paper. Okay, so you are able to copy it from here. Okay, and if you paste it here, it shows you this sequence. And you right click it and say create primer, and then it lets you make that gray region and name it.
Okay.
So I did the same thing for another paper here. So I select the forward and reverse that they use and right click and say link primers. So when you link these two, it lets you look at heterodimers. Otherwise, it will only look at one primer individually and it only looks at the homodimer.
Okay.
So now in the pairs, the second pair has come up. So I'll click on that. And I'm not sure if this is a good one or not. So I named it T1, which is temporary. So the difference is also not too large. It's 0.4. Okay, and even these ones, they have no monomer hairpins. So that's a good thing. Let's just look at this structure. I think it's decent. If it had 4 Gs, I would be slightly concerned. Even if the energy was lower, that would like make me want to mess with it. So I will either delete the last 3 sequences or whatever. And then the heterodimer has 5 things. It has 3 GCs. So maybe we can delete the last 2 Gs. Let's look at that. Okay, so the guideline is that it should end in a G or C. That means the 3' end, that's where the polymerase will bind and extrapolate the thing. If it has stronger binding there, it's more likely the polymerase will amplify successfully. So if you end on a G or C, it's beneficial. It's called the GC clamp. But if you have too many GCs here, then it increases the chance that it binds somewhere else, where only the last 4 nucleotides bind. So if you can imagine only these 4 bind, the rest is like hanging around like a tail here. So sometimes you would amplify those kind of products too. Okay, so here what we could do is to open this primer here. Alright, so I will delete the last 2, G and A. Now you can see it's red colored because the 3' location is 6 generally, so you have to change it because you deleted 2, you reduce this by 2. Now this primer is shorter, so its melting temperature would have reduced. So the difference in melting temperature has increased. Right, so to avoid that, what we can do is to add an AGP. Let's say if we add 3 nucleotides, this has extended here. It's still 1.87. There's another G we can add. Okay, I think if we add one more nucleotide here, it's going from minus 1.7 to plus 2. So for now, this should be okay. So you click again on secondary structure. So it will recalculate with the new sequence that it modified. It still has 5 things that are binding, but now they are somewhere else in the middle. Right, so the previous 5 that were binding, I removed those so that it found some new things to bind to. So I guess we'll just not do this. So I think between minus 4 and minus 2 point something, I would prefer minus 2 point something. AGT. Yeah, I think this one is better than the previous one. So we can accept that this is a decent primer pair. The other thing is we don't have probes in the middle. Those papers did not use probe-based chemistry. So if you want to design primers from scratch, you would just copy. Okay, so once you modified anything, you have to click Save Primer Pair. Otherwise, if you close that, you lose that modification. Okay, so now if you want to design like primers from scratch without depending on literature, you copy a sequence region where you want your primers to bind to, and you go to this software called primer3. So if you search on Google, you'll find the first thing that comes up. So here you'll paste the sequence where you want your primers to bind to, and you'll pick that you want to pick a left primer, right primer, but you also want a probe. So you have to check the box in the middle. And then there are some options as to what length you want your sequence to be. So these are all preset for normal PCR. So they want a longer region. But for qPCR, we want a short region, around 70 to 100.
So this is, if you didn't have any primers, any literature to start from, this is what you would, this is how you would go about
Yeah.
finding your primers initially.
Generally, even if I find things from literature, I prefer to do this because each software that people use, they give slightly different combination of primers and probes.
Yeah.
They also use different thermodynamic parameters. So you can see there's a table of thermodynamic parameters. There are two papers, and if you use these two ones, it gives you different temperature for the same sequence.
Are there any newer ones? 90, 98 and
This is the latest one, 1998.
wow.
Okay, so here I have some preset things. So if I click on it, it lets me choose something that I've entered before. So what it's doing is it tries to find between the first range that you give, 70 to 120. If it fails there, it goes to 120 to 150, and the rest is the same as the default, starting from 150 to 250.
So this is the size of the primer or the
The
size of like
product, size of the
oh,
amplicon.
okay, okay.
So that would be the size between, okay, where is our primers?
Yeah, yeah. Is
Yeah,
there,
so that would be from here to here all the way.
yeah. So do you
That will
generally
be.
want that to be smaller?
I think for qPCR, it should be as small as possible. If it's a little longer than usual, you would have to increase your step where you do the polymerase amplification. So I've designed all the primers in such a way that we can multiplex all of these and have used the same time and temperature for everything in qPCR. And when you multiplex, there's one more thing that we didn't really look at, but if this primer for the S gene is binding to the previous N1 and 2 primers that we're using, that could also be an issue. It's a lot more tedious to look at all those combinations. So those ones you will not use Benchling, but you'll use third-party software. So you can say primer dimer check. Okay, so there's different software from different companies. You can use any of those.
Okay.
So generally, they all have slightly different variations of those thermodynamic algorithms I've showed you. Also, even within this algorithm, there's a thing for, so it takes into account how much sodium is in your reaction buffer, which we don't quite know because the biorad doesn't tell us what exactly is in the composition. So for now, we just ignore those things. But if you want to be really accurate, if you change the amount of magnesium or sodium in your buffer, then your binding changes because those ions will interact with the primers and the probe as well. They'll bind to DNA.
Okay.
Yeah, so we don't worry about any of those things as of now. So first thing I do is to change this length, and then there's an optimal table of primers. So the what length you want, 18 to 23 is the range, that's default, and there's a melting temperature range. For probe, the range that we use, it's slightly higher than the primers. So the reason for that is we want the probe to bind first. So the mechanism in which it works is, let's say the probe is sitting here. So the probe starts here. So if the probe binds first, then once the primer binds, immediately the polymerase comes here and it starts adding more nucleotides. As it encounters the probe as a roadblock, it chews up the starting from the 5 end. It chews up all the nucleotides and then releases the fluorescence. So if the probe doesn't bind quickly enough, the polymerase will finish amplifying this, and then the probe cannot bind in that cycle.
Okay.
So you design it to be slightly longer, 20, 25, 30. And also the melting temperature should be 5 to 7 degrees higher than the primer. So for the primer, it was optimum was 60. Here I chose 65. Go back to the primer, you will find, yeah, 57, 59, 62, right? So optimum was 59. Here I chose 69.
Okay.
Then you just click on pick primers and it will pick. It will show you in the figure here where the primer binds. So the probe can either be close, it can be close to one of the primers, but it should be on the same strand as the primer that it's close to.
Okay.
So you can leave a gap of 3 to 5 nucleotides here. I think there's like a thing that I have. If you're designing primers in the future, I can send you this lab note. I can give you access to my lab notebook, or I can also send you a PDF of this thing. So I read some guidelines from IDT the first time I was designing it.
Okay.
And they recommend that there should be a distance of 4 to 50 between primer and probe, and probe should be 66 to 8 degrees higher than the primer. So all these things I just got from some document from a company.
Okay.
All right, so we'll just copy this region from here, or we can copy it from, there's a thing above. So the best primer is on the top, so you can just copy from here and do the same thing that we did before, just search. So Control F I'm doing, it opens that search box, and I just paste it here. So I already made the primers for these two. I haven't done the probe. So the probe is called internal oligo. That's the technical name for it. So you can notice it's longer than the primers. It also binds at a higher temperature. Instead of 58, it's 63. But so the hairpin that this calculates, it is showing that there's a probability of forming hairpins. Hairpin is when the monomer binds onto itself. So we'll just look at it on Benchling because it's more visual. This is interesting. So it's binding on the top strand. So I'll make this a forward orientation, and I will name it T2 probe. So all the temporary ones I'm putting it in an archive folder. Once I finalize them, I'll bring it into the main folder.
Okay. So I'm sorry. What did you say about which strand you want the probe to be on? You want it to be on?
Yeah. So the probe can either be on the forward strand here, or
Yeah.
it can also be on the bottom strand here. It
Yeah.
won't make a much difference.
Oh, okay. Okay.
So generally, if it's here, we want it to be closer to this primer. But I think from the document, it says the primer probe distance should be 4 to 50, and it's well within it. So this thing distance is from here to here.
Okay. Yeah.
That's 12 nucleotides. I think it doesn't make much difference either way.
Yeah.
If it's too close, basically the polymerase will not be able to bind to the primer next to the primer. There'll be a steric hindrance.
Right. Okay.
That's the only thing. Okay, so we'll select these two things and show primer pair. Okay, so it's already paired up. So I'll go to the primer pair first, then do the quality check. Energies all seem reasonable. This structure looks okay to me. Nothing continuous binding. These energy looks higher. It's 330 now, right? There's also some delivery thing, right? Deliveries are going to be closed next week or something, or starting tomorrow.
I saw an email. I didn't read it, but there's something
Yeah,
about
so I'm trying
deliveries.
to order earliest, so we have this ready to
Okay.
go. So the primers look okay. I won't be too stringent now because we don't have time.
Yeah.
So for the probe, you'll right click it and say oligo analysis, check secondary structure. And so this is showing for only one nucleotide, only one oligonucleotide. Okay. And so the thermodynamic energy, it will also change depending on which temperature you put into your equation.
Hmm.
And we use 60 for our elongation temperature and the binding. Both are happening simultaneously.
Right.
So this energy looks problematic to me. Okay, you can see why.
Oh, yeah.
So primer 3 doesn't always do a good job. So I never trusted blindly.
Yeah.
And so you can see that it also had a high energy here.
Yeah.
So it makes sense that it's bad. But I don't know exactly how this is being calculated. It seems like all of the candidates has are bad except this one. So we'll try out this. So these are the other candidates, and they have some ranking mechanism depending on how close they are to the optimal parameters you have set. So
Yeah.
let's just try this one out. So the forward is here. We should find a way to save these things quickly. Maybe I'll paste. So this one was really bad, so I'll just label it just so we don't end up checking it again. Okay, we'll select the reverse and do the same. Yeah, so for the basic idea is clear, right? So we are using another third-party software
Yeah.
to do our thermodynamic estimations. Benchling also has, I don't know why it makes it this way. Benchling also has a primer design feature, but you don't have as much control. So
Mm.
in the first window that we saw, there were so many parameters to change, right?
Yeah.
So here there's a thing. You can go to primers and say create primers, and it also links to, you can say wizard here. It links to the same primer 3, but it has much fewer options. So they try to make it more user UI friendly. But for qPCR, it doesn't give me the best primers. Link primers, and then we have secondary structure. These look good, the energies look good, so I'm not bothering about checking the structure. Oligo analysis. Okay, this one looks good actually, and there's only 4 nucleotides. I think for probe, the main thing is the monomer thing that should be more stringent.
Okay.
Okay, so this one seems to be good. So what I'll do is,
Does this soft? What about binding? I guess there's lots of so combinations of like probe to reverse primer. Is that important or is that
yeah, that's a good point actually. I have never done those extra analysis,
mm,
but it will be good to do. Right now I have no time, so I'm just
I
skipping all of those.
don't worry. I don't,
Yeah.
so,
Okay, so one more thing we forgot to look at is the difference in Tn.
right.
Okay, so one more thing is here the temperature is 58 point something. The other one is also closed. The probe is 61. So that's a difference of only 3 degrees. It's not very ideal, but let's try one more candidate. If that doesn't work, then we'll just go with it. So I think this one has a higher difference and it also seems to have zeros in all. Let's try this one last. If you're recording this, you should send this to Esther actually. It might be useful for her.
Okay.
What I've been doing recently is just whatever people are recording, I've just been putting it on YouTube. So if anyone has the same question,
I could send this to you if you want.
yeah, that
You
would
could
be
put it
good.
on your YouTube. Your YouTubes.
Look at archive. So this one, you'll notice it's a little too close to the primer, so we'll delete the first 3 or 4 things here.
Okay.
One important thing about the probe, I haven't gone into that, but so the first nucleotide should not be a G. If it
Okay.
is a G, then the G has some structure where it reduces the fluorescence by a lot.
Interesting.
I don't know the exact chemistry behind it, but they say avoid a G.
Um, which side is the fluorescent? So the fluorescent or the fluorophore is on the
It is on the first nucleotide, on the
okay
5 prime.
5'.
Okay, this has a minus 5 point something. I think it's reasonable. But once we delete the first few things, we'll look at it again. This one looks a little higher. It's okay. Okay, so here we'll delete. Let's see. So right now there's a gap of 1. So if we delete 3 nucleotides, let's delete the GTTG. So the probe became a lot shorter now. So we should add ATTT. Increase this by 5. So I think this looks reasonable. Energy is a bit lower than usual, but we'll just go with it for now. Okay, so I have to remove the first two primers that I saved, so the names don't repeat. ಆಯ್ತಮ್ಮ. Hmm?
Do you ever? I just saw there's like there's a blast thing in that drop down menu. Do you ever like go compare these sequences to like the other, like so you get maybe you accidentally bind to something you didn't
Yeah.
need to bind to.
So if you have like a base database, you can check with that. So for in this case, I would probably check it with the rest of the SARS-CoV-2 genome. So you need to find the actual names of which to filter within the NCBI, and you can also check with the human genome so that it doesn't bind somewhere. I don't know if any of that is should be. I think there should be human DNA in wastewater, so we don't want it to bind there.
Yeah.
So that's
So
how we would check.
yeah, so you would like look at, okay. Hmm. How do you, so you just go to the blast?
Oh yeah, so we can try that out for this. We go to NCBI Blast. Actually, for checking primers, this is not the best one. So if you go back to Blast, so there's an option for doing primer blast. So within the Blast suite, it says design your primer specific to your template. So here you would paste your template, which is this whole thing. That's the desired template. You would paste the two primers here, so you can check both of them simultaneously. So for this one, you have to paste it in a 5' to 3' orientation. So if you paste copy directly, it pastes the wrong strand. So you have to do copy special reverse complement.
Hmm.
That's the only thing. We need to be careful which orientation we need for the particular software.
Yeah.
Again, so for this, we would add like a query organism. I think right now Homo sapiens is selected by default. Yeah, it's not straightforward to. We'll just leave human for now. Yeah. I remember saving it somewhere. I have like a tab for bioinformatics. Yeah. No. This will just take a while to run. So in the
Okay.
meantime, we'll just go ahead and order these. So the best way to do it is, I think for now we are not ordering probe. The probe just takes a week to get delivered, so we have no hurry on that one.
So what are you gonna use a die or something or?
Yeah, so we have
So,
to think about what dye to use. So I think the idea is to use N2, and instead of N1 we will use this one. So this will be like a separate multiplex reaction, and so with the ratio of S to N2 we can tell if it's from the actual COVID or is it from the vaccine. If
wait,
the vaccine has only the S gene, so it should increase the amount
right.
we're detecting.
So I'm sorry. So what are you going to use for a probe then? Or are you going to? Sorry, I just kind of.
So we will use the Vic
Okay.
dye, which was previously applied on N1, and so instead of doing a multiplex with N1 and 2, we will do multiplex with S and N2
Okay. But
for
so you
this.
said you weren't going to order the probe next week, though.
Yeah, I will order it after doing this, and there's another primer set I'm designing
Okay.
for doing a, so for cloning. That's a different thing.
I'd be interested to learn about cloning as well.
Sure, yeah. Just a minute. Let me take this call.
Okay.
Hello. The spam call.
Dang spam calls. It picked up again recently, at least for me.
So the primer blast just looks like this. You'll have a graphical overview. It shows you where your primers are binding, and I don't know what the template is. Looks like the whole human DNA. 3.8. Oh no, this is the actual sequence that we input. This is the SARS S gene. And these are the bindings that are happening to the human genome. So all the dots are places where it's matching. The place with the A or G, that's where it's a mismatch. It seems like it's binding reasonably to certain areas on the human DNA. So there are some other, so within primer 3, you can actually build this in where you give it the human genome and say pick the primers that don't bind to that, but it will just take a lot longer.
Yeah. So this one is you think is fine in regards to that?
I think it's reasonable. So I think the ones that are more concerning is the short product length, because if it's too long, then the amount of time we are giving it, it will make sure that this product never gets made.
Right.
But
Okay.
if it's short, as in 3 to 400, but it has 5 places where it's a mismatch. Also, we have the probe, right? So I hope the probe should be discriminating between
True.
these things. So for the first round, we are planning to use just a dye-based thing because the probe we will not get it by mid next week. Okay, so to order that, usually I save this result. Maybe let's save this. I'm only saving the first 5. And so I'll go to the Sigma Aldrich website. So here you can figure out where is the oligo order, custom DNA oligos. I just save a shortcut for this. So when I search for oligo, I directly go to this page. Okay, so we'll just copy paste the sequences here. So if you go to primers, it lets you select the individual ones. So we'll just select these two and we'll go to copy to. So there was a sheet where I was saving these before I ordered them. So the first time when I order anything, I order the smallest quantity because it delivers the fastest.
Hmm.
So vendor is Sigma. I'm just keeping a record here because we've been ordering so many things lately just to see which stuff we got. So it just needs those three things for a shortcut ordering. If you directly copy paste, you won't have the 0.025. I think it's supposed to be this. Maybe it will throw an error. Oh, okay, this is the smallest quantity. Let's also order the thing from literature. We'll rename it later. Lauren likes to have primers from literature, that way you can compare. I'll have to name this something else. You need to have this checked, otherwise it makes an error. Yeah, so generally these different names, sometimes when I put them in the box, it's hard to write it on the tiny tubes, right? So sometimes people get confused. They use the wrong primer with the wrong probe.
11 cents per probe, looks like, or per base.
Yeah, we are 4 minutes before time. That's good. Generally, I order 1 minute before 4 or 5 p.m.